We are recording. If you're watching this video, so not you guys who are here right now, not the live stream, but if you're watching this video at a later date, you could probably skip ahead by about 4 minutes. But then again, since we have 4 minutes before the class officially starts, I'll give you guys reminders that I've been saying throughout the semester because I wouldn't want to give new important information before a class officially started. So I'm just going to fill this space in with some good reminders. So again, independent work is due at the end of the semester. You should start working on it now. It's easy. I mean, depending on how you operate, but in my opinion, for most people, it's easy to write short papers, right? Think of a question about biology, go find the answer and send me a paper. It could be, it might just be a few sentences, and that's okay. And likewise, like I keep saying throughout my lectures, you could also, something I say or something that the textbook says, you could copy it, paste it into your paper and say, hey, the textbook says this, and then go to a source and look it up from some other source and copy it from your source and paste it. Don't forget to use quotation marks and say, this source says this, and then compare and contrast what the two say. Beyond that, if you need any ideas of papers to write, let me know. I can help you come up with ideas as well. But I know it doesn't seem like it, but you guys are running out of time. And for those of you who have turned in papers, I wish you could tell everybody it's not as straightforward as you think. So if you wait until the last weekend to turn in these papers and you did it wrong and you get 0 credit, there's nothing I can do. You won't have time to make any corrections. So like I've been saying from day one, please at least turn in one paper if you haven't already. Turn in one paper. Make it a goal this weekend to turn in one paper so you can see how I grade. Please. It's a 100-point assignment. So, that's a whole letter grade. What are some other, reminders instead of announcements, things that I've been saying all semester. Make sure you're on lab. You arrive to lab on time. So this Wednesday, for example, I will lock the door at 10:01 a.m. All right. So if you're not there before 10:01, then you can't come to lab. Let's see, what are some other things? Attendance. Well, I'll wait till we officially start before I make that a reminder because it is important. What else? Coming to me for help. Please come to me for help. Whether you're doing an assignment, if you need help while you're doing the assignment, meet me online, or if you have to, meet me in the office. The reason I don't like meeting on the office is because all the assignments are completed online. So if you were to meet me in the office, which is fine, it would just be like you breathing on, standing behind me, looking at my screen, which I find awkward, or vice versa, me standing behind you, looking at your screen. So this is why I prefer to meet online. But yeah, let me help you, right? Let me help you complete assignments. Let me look over your assignment before you turn it in to make sure you didn't miss anything and you'll at least get some kind of credit. Meet me after I've graded an assignment, so I can give you feedback and then a 5% boost to that grade, the grade for that assignment. Yeah, so I think that's just the usual reminders. Read your emails, read the announcements, the usual stuff. And it's almost 8, so once it's 8, then I'll do the more important announcements / reminders. All right, it's 8. So the more important announcements and reminders before we get started, I want to let you know that today's attendance will be like usual. So I'm going to give you a bunch of questions, right? So you're going to send in a bunch of attendance words. And then the last attendance word is going to be me asking you to ask me a question. So as I'm going through today's lecture, make sure you think of start thinking about that. Maybe have something to write with. So if you have a question, go ahead and write it down. That way you can send it before 9. You have to send these letters or these words in before 9 a.m. And again, some people still aren't doing it. So I don't know if you're not paying attention. You are not considered present unless you send in those words. So just because you're logged in doesn't mean you're considered present. And then the opposite, like I said during lab on Wednesday when we met online, don't give your buddies the words and have them just have them send the words in and not log in. Right? I know who's logged in and who's not. I get an automatic report from Google when we shut down, so that's considered cheating. Don't do that. What are some other announcements and reminders? So you've already got the email. If you were not in lab on Wednesday and or you haven't done the pre-lab, then you need to get that done by the end of today. And again, let me help you if you need it. I told you a couple of weeks ago that I'm no longer going to keep people out of lab for not doing pre-labs and stuff like that. So if you have not done, if you're not present and you haven't done the things you need to do by Friday, you can still come to lab on Wednesday, but you can't do any of the experiments. You can just watch, walk around and ask questions, but you can't do the experiments, which might sound nice, but if that's the case, you won't get full credit for your, for your lab. So make sure you get that done. Let me help you. And then the other thing I want to remind you, because still very few people have taken me up on this offer, you can still redo exam 1. There's nothing to lose. The retake is untimed. The retake is open book. The retake is, again, you take it as many times as you want until you're satisfied with your grade. And then once you're satisfied with your grade, send me an email and say, hey, can you update my grade? Right. So I don't know why no one's taking me up on this, but that offer ends when we take the next exam. And speaking of the next exam, I'm going to open it pretty soon, but it only covers the stuff that we just talked about. Right. So it's going to talk about we're in chapter 5 now. So chapter 4 and 5, I believe. So it's all about, when it comes to all the properties of life, this exam is going to be almost entirely about energy, right? So when we talked about cellular respiration, right, that's getting energy from breaking down glucose. Now we're talking about photosynthesis, which is using the solar energy to create that chemical energy of glucose. There's going to be questions about enzymes and stuff like that. So long story short, this chapter that we're covering right now, this is the last chapter that's going to be on the exam. So even though it doesn't close, the exam doesn't close until like mid-March. You've got plenty of time to take it. But when I do open it, I'll let you know. But yeah, so after we finish this chapter 5, we're going to start new information. But that new information for that next chapter, there will not be on exam 2. So again, chapter 5 is the last of the information that you'll need for exam 2. So any questions before we get started? All right. This is where we left off. This is from the old textbook. The old textbook has something called, for each chapter, they had something called the process of science. And I erased it from most of the PowerPoints, but I kept it on this one so you can read about it. I'm not going to ask you any questions about this portion of the PowerPoint. Again, no questions on the process of science. But it is interesting. It's a little bit like a different version of what some of you guys are going to do in lab. Because what some of you are going to do in lab is see which color of light is best for photosynthesis. The way this person did it was they shined a prism, right? So they shined a rainbow on these algae, and then they had these bacteria that are aerobic, right? So they gravitate towards oxygen. And you can see here that there's a lot of them over here and a lot of them over here, meaning this color, these are the colors that are really good for photosynthesis, and these middle ones are not. So, excuse me, let me get this going really quickly. There we go. And the next slide. If you download the PowerPoint, you can click this and watch a little animation that talks a little bit what we've been talking about, the deals with pigments and lights. One of the questions that you asked when you sent in the attendance words on Wednesday was a good question. Actually, I'll back up too. And you don't need to know this for the exam, but someone had asked, why is red light the least powerful, the least energetic light, and why is blue light the most energetic light? And again, it comes back to this concept, and I wish we were in person. This will make a lot more sense. You see, it's in waves. Now imagine if I was holding a rope on one end, right? And then you were holding a rope on the other end. I'm a great artist, right? I should have double majored in biology and art. Anyway, now imagine like moving this up and down to make, if you move a rope up and down, you're going to get those waves. Now imagine the faster you do it, the shorter that wavelength is going to be, right? So this is why you can think of it as the same concept, right? So the faster it's moving, the shorter the wavelength is going to be, and the shorter the wavelengths is, the higher the energy, right? Meanwhile, the more distance between the wavelengths, the less energy it is. So that answers that question that you guys, a couple of you had asked in the email, but that will not be on the exam. So my first question for attendance is, will I ask you a question on the exam about why blue is higher energy than red? Yes or no? So the first word for attendance, the first official word for attendance is yes or no. Any questions before we move forward? All right. Now I've got to go through all this stuff we've already skipped. There we go. All right. So to kind of remind you what we've talked about so far, because I know some people weren't present on Monday and or Wednesday. First, we did the basics of photosynthesis. So we've already talked about the big overview, right? At this point, you already know the big picture. Now we're working our way into getting into more details, right? To talk about these two stages in more detail. But before we get there, there's some background information you need to know. And the one we just finished was the nature of sunlight, right? So you now know a little bit more about that, about how light works. So let's move forward and talk about some chloroplast pigments. And I've already mentioned this, but I'm mentioning it again. Selective absorption of light by leaves is why they appear green. So I mentioned that earlier, right? The reason leaves look green to us, or anything that's green, but obviously in the conversation of photosynthesis, leaves are what we're concerned with. But the reason they look green is because they are absorbing all the colors, right? So white light, for example, sunlight includes all the colors. So they're absorbing that color and absorbing all the colors other than green. And that is the one color that's being reflected back. So that is why things look that way. And again, I wish we were in person because I could use some other examples and point at some things that we could all look at. But, if you're wearing, I don't know, a blue shirt right now, the reason that shirt is blue is because the light is hitting it. It's absorbing all the colors other than blue. And blue is the one that is being reflected back. And then one last example I'm pretty sure you're aware, if you wear a black shirt out on a sunny, hot day, it's going to make you even hotter. The reason that is because of this bullet point right here. Well, first of all, black means it's not reflecting anything technically, right? Black means it's absorbing all the colors, which is why it appears black to us. So think about that. You get hot, and that's the reason you get hot more in a black shirt than any other colored shirt, because it's absorbing all the colors. And that brings us to this bullet point. Energy, as we know from earlier in the last chapter, and you need to know this for the exam, energy cannot be created and it cannot be destroyed, right? So these pigments are absorbing this energy, right? This light energy, then that has to be converted to other forms, right? It's not just going to disappear. And using the black shirt as an example, not that I would test you on the question about a black shirt, but that's why it's so hot, right? So the electrons and the pigments in your shirt get excited, right? They are then high energy electrons, but they can't stay that way. It's a very unstable state. So then they drop that energy. And when they drop that energy, they drop it in the form of heat, right? And that's why you get hot. Just like the high energy electrons from respiration. If you remember, right, we said, I keep using the word high energy electrons, just not regular old electrons, but high energy electrons. They're high energy in respiration from the last chapter. And they go from protein to protein and they're giving off their energy. And in that case, instead of giving off their energy as heat, right, in that case, the energy is used to pump hydrogen ions against a concentration gradient. But that being said, let's talk a little bit more about this bullet point right here. But before I do, are there any questions about this slide? Okay. Yeah. So again, this is what we've said. What I've said. It's just visualized. It's a picture from the old textbook, right? So we're looking at leaves. We're kind of looking at an animation of inside the leaf, right? So we're looking at this one chloroplast. Of course, there's a lot of chloroplasts inside of a single leaf. Excuse me, single plant cell. But we're looking at just this one. This light, this white light, is hitting the chloroplast, right? And it's absorbing all the colors except green. Green is the one bouncing off, right? And that's why it appears green to us. So it's everything we just said. So any questions about that concept? All right. So here's some new information, and this might be on the exam. So I'll put a question mark next to this. So we keep saying chlorophyll, right? Chlorophyll, chlorophyll, chlorophyll. That's the pigment that we use. Turns out there's actually different types of chlorophyll. And not only that, but there's also other pigments that we need to be aware of. So let's talk about them. Chlorophyll A. This is the one that participates. Man, I can't talk today. Participates directly in the light reactions. And to remind you, photosynthesis is broken down into two stages. There's the light reactions, and there's the Calvin cycle, right? So we're talking about light reactions. Obviously, it's in the name, right? Anyway, this is the one that participates directly in the light reactions, and it absorbs mainly blue, violet, and red light. I'm putting an X to that because if I ask any questions about these different pigments, if anything, you would just need to know this about chlorophyll A. It's the main one. It's the one that participates directly in the light reactions. I'm not going to ask you which colors it absorbs, but it is worth noting, as you'll see it when we look at these other ones, they all absorb. They have their own colors that they absorb, which makes it, it's a good team, right? Because again, white light is all the colors. So, it's good to have different pigments that absorb different colors. Imagine if that was like the next stage of evolution. Imagine if plants had some other pigment that also absorbed green light, right? That would be crazy. They wouldn't look the same to us anymore, but they would also be a lot more productive, right? They'd be really churning out some photosynthesis if they were absorbing more energy from the sun. Anyway, any questions about chlorophyll A before we talk about chlorophyll B? All right, chlorophyll B. This absorbs mainly blue and orange light. And as you might imagine, like I just said for chlorophyll A, I'm not going to ask you that. If anything, I would ask you this: like, what does it do? Or more than likely, the question would be, which which of the following conveys absorbed energy to chlorophyll A? Right? And the answers, your choices would be chlorophyll A, which is obviously not the correct answer, chlorophyll B, or carotenoids. But I'm definitely not going to ask you about the colors of light. So, any questions about chlorophyll B? All right, next are the carotenoids. These absorb mainly blue-green light. Again, put an X through here. I'm not going to ask you about that. And what their purpose is, and again, this is what I might ask if anything, they absorb and dissipate excessive light energy that might damage the chlorophyll, right? So, in a sense, I mean, yeah, it is. Yeah, let's just keep it at that. Yeah, so you could think of it almost like sunblock for your for the leaves, right? Because it's protecting the chlorophyll. Um, yeah. So, any questions about carotenoids? All right. So the next, if you have a question, I can't if you're typing something or raising your hand, I can't see it. So you could just unmute yourself and ask the question if you want.
Okay. Could you
Oh,
could
good.
you please could you please go over the chlorophyll B again, please?
Yes. Basically, chlorophyll B, it absorbs energy, and then it directs the energy to chlorophyll A. And that's basically it. And anything that you need to know about chlorophyll B, if I ask, which I probably won't, you need to know the chlorophyll B's purpose is to kind of absorb that solar energy, but then pass that energy on to chlorophyll A. So chlorophyll A is the one that's doing all the work.
So you said chlorophyll A it participates in the light reaction, and does the chlorophyll B participates in that as well?
Not directly. And that'll make more sense when we talk about the light reactions. But for now, just know that it's chlorophyll A that's directly participating in it. Chlorophyll B is just passing that energy on to chlorophyll A. And again, the carotenoids are there just to protect the chlorophylls. Good question. Anything else?
Thank you.
Thank you. Yeah, definitely ask questions, guys, because I've been teaching this for so long. It just like I have this memorized and it just makes sense to me. So I need to know when something doesn't make sense to you. So thank you very much for asking questions. But all right. So the next word for attendance is to tell me this. On the exam, if I ask any questions about chlorophyll A, chlorophyll B, or carotenoids, will I ask you any questions about the color that they absorb? So, for example, you can see on the screen right here, carotenoids absorb mainly blue-green light, right? They all absorb different colors. Will that be one of the questions? Will I ask you what colors they absorb? That is the next word for attendance. So let's talk about some of these carotenoids. Not that I'm going to ask you, but I feel like it's kind of interesting. You have beta carotene. This is a bright orange-ish red pigment that you find in pumpkins and sweet potatoes and carrots. And then your body converts that to vitamin A. So that's why it's good for you. That's why it's important. That's an independent work topic you could look into, right? How does your body convert beta carotene into vitamin A? Or what does vitamin A do for your body, right? And or where else can we get beta carotene other than pumpkins, sweet potatoes, and carrots, right? Those are all things you could look up for independent work. So that's beta carotene. Any questions about beta carotene? All right, the next one, and we're not even talking about all the carotenoids. These are only some of them. The next one we're talking about is lycopene. This is a bright red pigment you can find in tomatoes and watermelons and red peppers. And it's an antioxidant, right? And it's being studied for potential anti-cancer properties, which again brings up other independent work topics. Where else can we find lycopene other than tomatoes, watermelons, and red peppers? What is an antioxidant, right? Think questions like that. But again, big ol' X through here, meaning I'm not going to ask any questions about these things, which means I'm also going to move forward instead of taking your questions about these things. So we're still talking about these carotenoids. Again, not important for the exam, but relevant to your everyday life. This is where we get our fall colors from, partially, right? It's because of these carotenoids. So what happens is when the temperature changes, and not just the temperature, it's also like the amount of sunlight that hits every day. Because as you know, hopefully you know this, as we move towards, once we hit fall, then every day gets a little bit shorter until we hit, the middle of winter. So yeah, that has something to do with it. But anyway, yeah. So when you look at green leaves, of course, there's no leaves out right now, but if you were to look in the summertime and see a bunch of green leaves, all these fall colors are still there. They're just masked by the chlorophyll. And what happens, again, like I said earlier, with the change in weather, that chlorophyll goes away, and that allows us to see these colors that were always there, these carotenoids. Which brings up another independent work topic. When I said the chlorophyll goes away, well, where does it go? Right. So we know not only can energy not be created or destroyed, but neither can matter. Right. So what happens to those chlorophyll molecules? Are they just evaporated? Are they broken down? What happens to them? You can write about that for independent work. Any questions about this relatively unimportant slide? Okay. Here's a little graph. I'm not going to ask you any questions about this, but it shows which lights are absorbed by chlorophyll A, right? You can see which lights are colors, are absorbed by chlorophyll B. You can see which colors are absorbed by the carotenoids. Yeah, so I'm not going to ask you this, but there you go. If anything, it's like a 99% chance that I'm not going to do this because it's not on the exam currently, and I don't feel like I'm going to add it. But if anything, I might give you this graph, which is not your textbook, by the way. This is the old textbook. I might give you this graph and ask you questions about it for you to decipher it. Like I might say, which of the pigments does best at absorbing, I don't know, 680 nanometers, right? That looks like about 680 to me, if I had to guess, right? So you look and be like, okay, definitely this one. And then you look at the key and be like, okay, that's chlorophyll A, right? Or I might say, which one does best at 650? Or which one does best at, well, I probably wouldn't ask questions about this because they're so close, but yeah. So yeah, if anything, like I said, I might give you this graph and ask you a question, ask you to decipher it. Now that I think about it, maybe not for this semester. So I'm kind of preaching to the choir here, but I might start doing that some more. I think being able to decipher a graph is a very important thing, especially these days. And it's not just about biology, right? Even politically, with all the stuff going on, right now politically, to be able to decipher a graph that's showing, I don't know, unemployment rate or inflation rate or measles outbreaks in Texas, right? All these things would be, be important to know. Anyway, any questions about this slide? I mean, yeah, slide, graph, whatever, before we move forward. All right. Finally, now we have enough background information where we can dig deep into the 2 stages of photosynthesis. And I'll remind you, for the exam, the questions are going to be me saying, in which stage does this happen? In which stage does that happen? In which stage is this used? In which stage is this produced? So as I'm talking about that, keep in mind that that's how a large majority of the questions are going to go. And like I said on Wednesday, it's a multiple choice exam, and the only 2 correct answers for a photosynthesis question would be light reactions or the Calvin cycle. But again, it's multiple choice, so there's going to be more than 2 choices, right? There's going to be other nonsense on there. So don't choose the nonsense. If you get the question wrong, but you at least guess something from photosynthesis and none of the nonsense, then you will get at least partial credit. So again, when you take take the exam and a question asks about photosynthesis, you're not going to use glycolysis will not be an answer because that's from respiration. Electric citric acid cycle will not be an option because that's from respiration. Mitosis, which is what we're going to learn about on the next chapter, that will not be an option, right? And not a correct answer. So yeah, that being said, let's jump into it. As you already know, light behaves as waves. That's not necessarily important in my opinion. I might ask you that again. We just talked earlier about how this is why blue energy is high, blue light is high energy, right? Just imagine again, holding a rope and moving it up and down really quickly, right? So that's how you can think of it as waves. But it also acts as discrete packets of energy called photons. And here's what you need to know about photons. You just need to know that's just the packet of energy, of light energy. I'm not going to test you on that word, but I'm going to use that word moving forward. So you need to know what I'm talking about. And the question, there might be questions on the exam that use that word, but, no questions asking you what it is. So again, I'm not going to ask you the definition, but you'll need to know what it means. Otherwise you won't understand that question. So again, this is like light energy, right? It's a fixed quantity of light energy. For example, if you have, a weak lamp versus a very strong lamp, right? The very strong lamp is giving you more photons, more fixed quantities of light energy. The weak lamp would be giving you relatively fewer photons, right? Fewer fixed quantities of light energy. And again, like we have said earlier, not that you need to know this, but the shorter the wavelength, the greater the energy of the photon, right? So again, blue light, very, very energetic. Red light, relatively very, unenergetic. So again, as far as the exam is concerned, this slide is not very important. But yeah, you just need to know what photons are because I'm going to use the word. In case you're wondering what this picture is about, this is why I don't know if you guys know, but I'm sure you do. Sunblock, it's supposed to block out UV light, ultraviolet. So that's even more powerful. I don't know if you remember the graph, but we have red here and blue here, right? And again, that's least energy. That's more energy, right? So this is where ultraviolet is. But anyway, let's move forward. So let's talk about what happens. And I mentioned this earlier when I was talking about black shirts. So we're coming back to this concept. What happens when a pigment molecule absorbs a photon? What happens is, like I said previously, now you can see it in writing, the electrons in those pigments get excited. They gain that energy, right? Because energy cannot be created or destroyed. So it is now in an excited state. But this excited state is very unstable. So usually what happens is they'll lose that excess energy almost immediately, and fall back down to their ground state. And the ground state is basically the opposite of excited state. Ground state, just a regular old electron, right? Most pigments, again, like using the black shirt as an example, they release that energy as heat energy, right? And again, this is from, I think the old textbook and maybe yours is the only reason I'm including it in this, but what I'm about to tell you is not important. It is slightly interesting, I guess. So let's talk about it. Sometimes what happens when a pigment gets excited, right? Sometimes as it loses that excitement, it releases that energy as light as opposed to heat. Not that you need to know this, but yeah. And here's a picture of what we're talking about, right? So generally what happens, again, we have the light from the sun, well, from any light source, right? So these photons come and they hit the chlorophyll molecule, particularly chlorophyll A, and we'll talk about that here in a second. That electron gets very excited. It can't stay that way. So generally what would happen, and this is not what actually happens in photosynthesis. So when I think about it, it's kind of a stupid, stupid picture. But what would normally happen when in most pigments other than chlorophyll is the electron would get excited. And then again, it can't stay that way. So it drops back down and it releases that energy, usually as heat, sometimes as light. And then there's a third thing that we're going to talk about later in this chapter. And that third thing is actually the more important thing. But yeah, I guess that the main concept here is photons excite electrons, and electrons cannot keep that excitement, right? So they absorb that energy and become excited, and then they quickly lose that energy and become, back down to the ground state. I would ask if there's any questions, but I don't want to do it yet because I want to make sure we get the entire concept out there, and then I'll take your questions. Again, completely useless, and this is from your old textbook. But yes, sometimes, like that picture I just showed you, yes, it releases heat energy sometimes, and sometimes it releases light energy. And these glow sticks are a great example, or anything that glows. That's how that works. You shine a light on it. Well, forget the glow sticks for a second. When you think about something that glows and it's not a glow stick, what's happening is you're shining a light on it. You're exciting those electrons. It just so happens that chemical is very good at maintaining that excitement for a little bit longer time, and it lets it slowly give off that energy, right? And this is why things can glow for a while, and then you have to recharge them again by putting them in the light. And then of course there's these glow sticks, and again, this is unimportant for the exam or for understanding anything in the semester, but I might as well tell you about it. I don't know if you've ever used one, but they don't glow until you break them, right? You crack them and then the chemicals mix. So what you're seeing here is these electrons are excited not from photons, but they're excited from a chemical reaction. And again, they can't stay at that excited state. So as they lose that energy, that energy is then released as light. Again, for the exam, that's not important. So I won't even take questions on that. I'll just keep moving forward. All right. So in the thylakoid membrane of the chloroplast, chlorophyll molecules are organized with each other and other molecules into something called photosystems. I'm not going to ask you what a photosystem is, but I will be using that word. So you're going to need to understand what it means. And that'll make more sense here in the next couple of slides. But each photosystem has a cluster of a few hundred pigment molecules. And again, I'm not going to ask you that, but here's what a photosystem looks like. Right? So again, inside the chloroplast, we're looking at this thylakoid membrane and we're zooming in, right? And look, this should look familiar, phospholipid bilayer, just like a cell has, right? But anyway, embedded in that membrane is these things called photosystems, photosystems. And yeah, there's just hundreds of chlorophyll pigments and other pigments embedded in that photosystem. We haven't talked about what they've done yet, and I will, but this is what they are. I'm probably not going to ask you the fact that they're on the thylakoid membrane, but you definitely need to know that all of this is taking place in the chloroplast. Everything I'm telling you about photosynthesis happens in the chloroplast. Now, we could get a little bit more specific and say in the thylakoid membrane of the chloroplast, but for now, big picture, just know that all of this is happening in the chloroplast. And again, we don't know what these photosystems do yet. I'm about to tell you, but before I move forward and tell you what they do you have any questions about them? All right, and again, this picture not again, I haven't told you this yet. This particular picture is not in your textbook. This is from the old textbook. So, if it's a good visualization for you, you might want to download this PowerPoint. All right. So if there's no, I won't ask you that. All right, let's move forward. So let's talk about what they do, right? This cluster of pigments is basically it's the light gathering antenna. You could almost think of it as a solar panel, right? So I used this analogy earlier in the week where I said you can think of Calvin cycle. That's the thing that actually builds the glucose molecules, which is what the whole purpose of photosynthesis is, right? To make that glucose. You could think of that as the factory that builds it, right? And the light reactions, I said earlier in the week, are basically the power plant, right? It's the thing providing the energy so the factory can do its job. So now I'm getting even more specific with that metaphor, and I'm saying that the light reactions, right, specifically the photosystems, which which are used in the light reactions, those are basically like solar antennas, right? They're absorbing that light energy. The photon strikes the pigments, as we mentioned earlier in that picture I showed you, right? Or that one picture where it had a chlorophyll molecule and it had the sun, and it was, the light was hitting it, and then the electron got excited, right? And then the electron fell back down, right? This is what we're talking about. The energy from the photon, again, where the photon strikes the pigment, the energy from that, right, excites the electron. Well, let's talk about this first. Technically, the energy jumps from molecule to molecule, and then finally, at a certain point, it hits the reaction center, and the reaction center is finally where it happens, right? What I previously mentioned, this is where it happens. All that energy finally gets to the reaction center, and the reaction center consists of one chlorophyll A molecule. So that goes back to what we were talking about, right? So the rest of them were chlorophyll B, right? They, as far as we're concerned, and other pigments, right? So they're absorbing that energy, they're passing it around back and forth, and it's just bouncing around until finally it hits the reaction center, which is where the chlorophyll A is. And that chlorophyll A sits right next to something called the primary electron acceptor, which then excites that excited electron. So going back to this picture, again, the photons hit it, it's bouncing around a bunch, and then finally it hits the reaction center, which is where chlorophyll A is. That electron gets excited, but let's erase that, because unlike in that last picture, come on, I can't erase it. Anyway, unlike in the last picture, the electrons are not going to lose that energy. They're going to be caught by this thing called a primary electron acceptor. It's going to catch that excited electron. So again, big picture here. Excuse me, before I give you the big picture. And then now that the primary electron acceptor has that high energy electron, then it kind of moves on, and another team of molecules uses that trapped energy to make ATP and NADPH. And I will talk more about this on the next slide. So right now, I'm not going to ask if you have any questions about this, because I still need to explain it. But are there any questions about this in general other than here down, right? So I'm saying, are there any questions about this idea of the photon hitting the pigments and the energy bouncing around until it hits the reaction center, which is where chlorophyll A is? At that point, the energy gets excited, the electron gets excited, and it is captured by the primary electron acceptor. Are there any questions about that?
So this whole thing is happening right now in the light reaction, right?
Yes.
Okay.
Good question. Yes. Right now, this is all light reactions. And hopefully that'll be easy for you on the exam, because it's all in the name, like you said, right? Light reactions. So if it has anything to do with pigments, all right, that's going to be light reactions. If it has anything to do with electrons getting excited, right, that's getting excited from the photons, right? So that has to be light reactions. And like I said previously in the week, even though I'm about to talk about this bullet point on the next slide, I will mention again, one test question will definitely be, which of the two stages produces ATP and NADPH? And the answer is the light reactions, right? They produce the ATP that the Calvin cycle is going to use to build glucose. They produce this NADPH, right? So we can have these high energy electrons that can then be incorporated into that glucose on the Calvin cycle, right? So the Calvin cycle uses this stuff, but the light reactions produce ATP and NADPH. And I am going to talk about that in the upcoming slides. But the next word for attendance is the light reactions produce two things, NADPH and what else? That's your next word for attendance. The light reactions produce two things, NADPH and what else? All right, now let's talk about it. Again, with the pictures. This is a much better picture. So this is a picture of what we were talking about. So we've already saw this, right? Here's a chloroplast. We're zooming in. We're looking at this one thylakoid membrane. Here's this one photosystem. You know, there's a bunch of photosystems in those thylakoid membranes, but we're just looking at this one with all these different pigments. The photon hits one, right? It gets excited. That excitement just kind of bounces around and bounces around, bounces around until finally it gets to the reaction center, which is where chlorophyll A is, right? Then the electron in that chlorophyll A becomes excited, right? It gets into that excited state. But instead of coming back down from that excited state, it's captured by the primary electron acceptor in its excited state. And what I like about photosynthesis, or at least the stuff we're talking about now, is it's very much in the name, right? Primary electron acceptor. It kind of tells you what to do or what it does, right? It's like, so if I were to ask in the exam, you know, what is it that captures that first excited electron? The primary electron acceptor, right? It's right there in the name. And also, and I probably won't ask this, but if I did, you know, again, if I said which pigment is the one that donates that electron? Is it chlorophyll A, chlorophyll B, or chlorophyll or the carotenoids? And hopefully, you know, chlorophyll A, right? The first one listed. Hopefully that'll help you remember that that's the one that's in the reaction center. That's the one that's donating that electron. Because again, it is donating it. The primary electron acceptor, like I said in the last slide, and we're still going to talk about it in the upcoming slides, it steals those electrons, right? And it does something else with them. So it is taking those electrons. Those chlorophyll are giving up their electrons. And we'll talk about that too, because like I said when we were talking about respiration, electrons have to come from somewhere and they have to go somewhere. So when chlorophyll just donates these electrons, they have to be replaced. And we'll talk about that here in a second. So let's talk about this, right? Like I said in 2 slides ago, that last bullet point, I said we're going to talk about it. So here it is. We're going to talk about how the light reactions generate ATP and NADPH, right? And again, that's definitely a test question. Which of the 2 stages generate or make or produce ATP and NADPH? Light reactions. Now I'm about to explain exactly how it works, but most likely I'm not going to get that deep into it for the exam. But you definitely need to know that light reactions produce ATP and produce NADPH. And as you'll learn later, that ATP and those high energy electrons that are captured in the NADPH, they are used by the Calvin cycle. So don't let that confuse you on the exam, because ATP and NADPH are associated with both, but light reactions generate it. Calvin cycle uses it. So what I didn't tell you yet, we talked about photosystems. There's actually 2 photosystems that cooperate in the light reactions. So what's happening in the first one that we talked about anyway, is the photons the photons, this is what we've already talked about. The photons excite those electrons, right? They bounce around. Then they finally get to the reaction center, which is where chlorophyll A is. That electron becomes, you know, excited, right? And then it is then captured by the primary electron acceptor. This is things we've already said. But then, like I said earlier, before we got to this slide, the photosystem has to replace those electrons, right? Electrons have to come from somewhere. So the water, the chlorophyll A loses an electron. So where's it going to get replaced? And the answer is water. So again, that'll definitely be a test question. First of all, the 2 test questions, right? Because one test question will be, which of the 2 stages uses water? The light reactions or the Calvin cycle? And the answer is the light reactions. And hopefully this will make sense, because it's the water that's donating those electrons, right? And when that happens, we produce a molecule of water. And this should sound very familiar because I told you this was going to happen when we were talking about respiration, right? And respiration, I said we had these high energy electrons that came from glucose, right? And they were accepted by oxygen. And the process of doing that gave us water, right? And then I told you at the time, it's going to be the opposite for photosynthesis. And photosynthesis, the water is like, hey, here's the electron that's eventually going to be incorporated into glucose. But the process of doing that produces oxygen. And I told you for the exam, so obviously there's going to be two questions like this, one about photosynthesis and one about respiration. But you should associate these things together. Water, oxygen, and electrons, right? Those things should be associated, whether it's water giving up an electron and producing oxygen, or oxygen absorbing electron, like in respiration, and producing water. So are there any questions about this slide before we talk about the second photosystem? So still, again, we're still talking about that first photosystem that we talked about. We haven't moved forward. We just added more information. All right, then let's move forward. And here's a picture of what we're talking about. So again, this should look familiar, right? Photons hit the pigments, they get excited, blah, blah, blah. The excitement finally hits the reaction center where chlorophyll A is. That electron gets, technically 2 electrons get excited and then taken away by this primary electron acceptor. There we go. We've lost some electrons. We need more. Where do they come from? They come from water, and that produces oxygen. But I'll remind you, I'm not going to be asking you about like how many electrons, right? It won't be asking you that. It won't be asking you questions about how technically there's also 2 hydrogen protons involved in this reaction. None of that. Just know that in photosynthesis, the opposite of respiration, water is giving up electrons, and in the process, that produces oxygen. So any questions about this slide? Okay, so let's talk about the next photosystem. So again, right, here's where we left off. This is basically a story about the electrons, right? The electrons started from the water, then they went into chlorophyll, they got excited, and then they were taken away by the primary electron acceptor, right? So this is where we're leaving off. We've got this excited electron that has been taken by the primary electron acceptor, and now it's going to go into the second photosystem. Another good independent work topic if you want to look into it. I keep saying primary electron acceptor, but what is it? What exactly is the primary electron acceptor? Like, what kind of molecule is it? Is it multiple molecules? Right? Those are things you don't need to know for the exam, but you can look it up for independent work. So anyway, again, we have these energized electrons that we got from the first photosystem, right? That primary electron acceptor stole them, so to speak. It took it, and then it passes it down to the second photosystem where we have the electron transport chain. Not to confuse you, but that should sound familiar, right? That was the last stage of respiration. So here we have another one. We have another electron transport chain that basically, as far as we're concerned in this discussion, it works the same way. Because remember, like I said in the previous slides, the purpose of the light reactions is to produce ATP and to produce high energy electrons, right? And as you know from respiration, we make ATP through the electron transport chain. Like, that's the best way to make ATP is through the electron transport chain. So again, first photosystem is all about exciting that electron and capturing it. Second photosystem is all about using the energy from that excited electron to make ATP and to make NADPH, right? To make ATP because the Calvin cycle needs energy, and to make NADPH because the Calvin cycle also needs high energy electrons that it can incorporate into that glucose molecule that it makes. And again, that's the next stage. And again, the test question here is, which of the 2 stages produces ATP, light reactions? Which of the 2 stages produces NADPH, light reactions? Which of the 2 stages utilizes an electron transport chain, light reactions? And even though I'm probably not going to ask it, technically it could be a test question, which of the 2 stages uses NADPH, or excuse me, NADP plus? That would be the light reactions. Because again, I'm probably not going to go that deep in the exam, but technically, yeah, it's using NADP plus because that's the thing that accepts the electrons. And once it accepts the electrons, it becomes technically NADPH. So again, light reactions use NADP plus, but they produce NADPH. And you know what? I'm going to go ahead and tell you, I'm for sure about this. I'm not going to ask about that because I want to make things actually easier for you. So again, if you get a question about which produces NADPH, the answer is light reactions. If you ask which uses NADPH, the answer is Calvin cycle. So if I were to throw this in there, that would make things even more complicated because you'd be like, oh, wait a minute. NADP plus, which one uses NADP plus? Then you'd have to memorize the difference between NADP plus and NADPH. And I don't want to make things that hard for you. So any questions on this slide before we move forward? All right. So again, here's a picture of what we're talking about. The photons excite the electrons in the first reaction center. Boom, it's excited. The primary electron acceptor takes it, and then it sends it on its way to the electron transport chain, which, just like in respiration, makes ATP, right? So this high energy electrons going from protein to protein, giving out that, releasing its energy, and that energy is used to make ATP. Essentially the same exact way that it did in respiration. And we'll talk about that here in a second too. I'll refresh your memory. Then it gets here. And notice the drawing. This is not from your textbook. This is from the old textbook. I like it. Notice how the first photosystem is down here. The second photosystem is a little bit up here. This signifies the excitement of the electron, right? So here, the water, that's when it's its least excited stage. Then it gets very excited up here. Then it's starting to lose some excitement. Then it gets down to the second photosystem. And again, notice, not that I'm going to ask this on the exam, but it's lost a lot of its excitement, which is why it's lower. The reaction second photosystem is lower than the primary electron acceptor, but it hasn't lost all of its energy, which is why the second photosystem is higher than the first photosystem. Anyway, it gets to the second photosystem. Light, it's not showing it. Oh, yeah, it is. Light is still hitting this too, right? So now it's exciting these electrons again. And it gets accepted by this primary electron acceptor. Lucky for you, look at that. They're the same name, so you don't have to worry about differentiating between the two. And this time, when it's excited and captured, instead of making more ATP, it is then captured by the NADP plus, turning it into NADPH. And there we go. Now we have what we're really after, right? They've completed the light reactions have completed what they needed to do, what they're really after, which is ATP and then NADPH, right? That's the whole purpose of the light reactions, is to produce energy in the form of ATP and to produce high energy electrons in the form of NADPH. We still have a little bit more to talk about this. Hopefully we can finish this before the class is up. We're running out of time, but are there any questions about this slide? All right. I like this too. This is the same. This is like just the same concept drawn in a different way. The photon excites the electron, right? And we have the primary electron acceptor that's like, all right, well, since I'm up here and I've got all this potential energy, let me roll this thing down, and it can lose some of its potential energy in the form of kinetic energy, and that'll turn this wheel, and that'll produce ATP. And then it gets here, and the photon's like, all right, let me excite you again. Let me have another electron acceptor. It's like, well, I've got this excited electron. Let me put it in this bucket. And this bucket is called NADPH, right? So we have what we're after, ATP and NADPH. Again, well, you know what? Let's see. Yeah. So like I said in the previous slide, the ATP production, as far as we're concerned, is the same as it is in respiration, right? Because in both of them, we're using an electron transport chain. And that electron transport chain is pumping hydrogen ions across the membrane against the concentration gradient. And then it uses the enzyme ATP synthase, just like it does in respiration, to produce ATP. So the last word for attendance, other than you sending me a question, the last actual word for attendance is, what enzyme does photosynthesis use to make ATP? What is the name of the enzyme that photosynthesis uses to make ATP? And then beyond that, like I said earlier, you also need to send me one question about the material that I presented today. And that's it. You guys can log off. I'll be on for office hours. Yeah, That's it. Let me help you. If you need help, please, please, please let me help you. You don't have to go at this alone. Some of you really need help, and you're not taking me up on it.